So hi everyone, my name is Jessica Johnson. I often go by Jess. Yes, I'm a graduate student with Los Alamos, and I'm pursuing my master's degree through the University of New Mexico in biology. And I'm fortunate enough to work with Los Alamos in the environmental stewardship program. Let me just hide. Okay. So I wanted to present to you all about the research that I've been working on for the past 2 years with UNM. I've spent the last 10 years of my career working with small mammals. And so my master's thesis was studying the sensitivities of small mammals to drought in an arid desert ecosystem. So I really hope you enjoy learning about small mammals because I'm very passionate about them. And I'm going to go ahead and turn off my video for now during my presentation, and then I will pop back in at the end. Okay. So to begin, why are we interested in studying small mammals? In case you weren't aware, in the class Mammalia, the small mammals, including rodents, bats, and shrews, comprise 70% of the mammalian class. Because there's so many small mammals within the mammalian class, it would be exceptionally challenging for us to study every species of rodent, and because we know that they are sensitive to climate change or indicator species, it's important to study what we can using long-term existing data on small mammals, and this may provide valuable insight into several families and genre of the rodent community. Furthermore, realistically, small mammals are relatively easy to capture when compared to other orders of large animals, particularly carnivores that are often the dominant predator species in ecosystems. It can be very difficult to capture, it can be dangerous and expensive. So utilizing small mammals has provided ecologists with a tangible way to replicate studies over long periods of time and obtain quite a few data points. So a little bit about small mammals and their role in ecosystems. So my research is fortunate enough to contain a very large data set that studies the same community of small mammals in the same place and time. So our analysis of this community allows us to make generalizations about each of these species, regardless of location. Keystone species are defined as species that an ecosystem is dependent on, and should they be removed, the ecosystem would be drastically altered. And keystone status can be applied to small mammals, especially in ecosystems where their abundances are high. Small mammals play a very important role in ecosystem functioning, and a broader example of an important ecosystem function that small mammals can play would be in a dry, semi-desert grassland ecosystem, such as my study site. Rodent burrows can increase water storage, which is an important contribution stemming solely from rodent presence in this ecosystem. A more specific example of their ecosystem functioning was actually done at my study site through the Museum of Southwestern Biology at the University of New Mexico. In the 90s, there was an exclusion study that was conducted where rodents were removed from several locations at the Sevilleta. And rodents in this community typically like plant species with large seeds and eat more of those. So usually the seed size is smaller because of the small mammals. However, when they removed them over the 10-year period, there's a shift in the plant community to larger seeds because the small mammals were no longer there to overconsume the larger seeds and keep the seed size small. I wanted to go ahead and introduce you to my study site that I've been referencing. It's called the Sevilleta National Wildlife Refuge. It's about an hour south of Albuquerque, and it spans 230,000 acres and spreads across 4 different biomes. And biomes are ecosystems with distinct types of vegetation, animals, and microbes. So this landscape has a wide array of biological diversity, making it a really unique system to study. Specifically, there are 23 species of small mammals at the Sevilleta, which is highly diverse, and their populations are abundant. Of those 4 biomes, the 2 that were the main habitats for my study included the shrubland and grassland, which contain creosote shrub and black grama grass. for some more information about the Sevilleta National Wildlife Refuge, it transitioned from ranch ownership in the 1970s and was donated for conservation effort to the Nature Conservancy and then passed on to the U.S. Fish and Wildlife. And they grant research permits as part of their conservation goal, including a research permit with the University of New Mexico that operates the Long-Term Ecological Research Network, often referred to as the LTER, where I was fortunate enough to work for 2 years doing my research. Some of the longer protocols that the LTER conducts at the Sevilleta include a small mammal trapping protocol that I use for my research that's ongoing and has been going since 1989. Another long-term data set is the climate data collected from meteorological stations that has been going since 1988. So I wanted to just quickly mention the true importance of long-term data sets. It's a novel concept because there are very few studies that span this length of time monitoring the same system and organisms in the same place. Because of the novelty, it allows us to make accurate predictions and models for future populations with these increasing changes to climate rather than inferences or educated guesses. Not only can this type of analysis be used in this specific system with these specific taxa, but any long-term studies can also use similar modeling techniques to try and understand trends and make future predictions in their systems. And I also wanted to touch on the fact that the value of these programs is still ongoing. The LTR has been funded since the 80s, and now in the 2000s, 2010s, the NSF started funding NEON, which is the National Ecological Observatory Network, where I also was fortunate enough to work for 4 years. And while the LTR is studying specific systems like the Sevilleta, the arid desert grassland, NEON's goal is to provide long-term data sets on a continental scale. So one of the goals of the Sevilleta, the specific system that our LTR is focused on, is climate change and variability. Global climate change is very well known, particularly in the science and ecology community. And we know that climate drivers such as increased global air temperatures, altered precipitation patterns, and increased climate variability from year to year are drivers that are being greatly altered. And trying to tie these climate changes to organisms allow us to understand the implications. Species level analyses in particular can be applied to this community and give us a more in-depth analysis of these effects on individuals and their population size. The picture that I decided to show on this slide is a direct comparison of climate and its implications on a system. This is done with the Collins Lab through the University of New Mexico at the Sevilleta. And the experiment was done from the LTR because the LTR not only just studies observational systems, but they also use experiments to manipulate climate so we can make better predictions about what we anticipate will happen in the future. So this experiment was called Extreme Drought in Grasslands Experiment, or EDGE. And here they removed 66% of monsoon rain, and in just 3 years, there was extreme plant mortality. A little more about drought across the United States since the 1900s. On average, we're seeing that we're experiencing a drier climate and a more variable climate. And to understand these more variable climates, we have introduced SPEI, or an aridity and drought index, which stands for Standardized Precipitation Evapotranspiration Index. And as you can see, I'll use my laser pointer here. The Sevilleta sits right here in a hot spot of a drier climate and a more variable climate. So that also makes it a very unique system to study. Some 100-year trends that we're seeing at the Sevilleta in regards to climate. We are seeing that the Sevilleta is getting drier on average, and the climate variability is also increasing. The plot on the left, you can see that drought aridity index that I referenced, and the positive numbers reference the cooler or wetter years, with the hot and dry years represented by the negative values. And as you can see, the trend line dictates that we are getting drier, and then the plot on the right shows the variation in drought that is increasing over time. So in order for us to be able to tie these climate trends that we're seeing to our organisms and our systems, a useful way is to use functional traits. And functional traits are broadly described as affecting a species' survival, development, growth, and reproduction. So we wanted to know if these traits can create adaptations to make species more resistant to climate change or more vulnerable. And a good example of research that's already been used in ecology to answer this prominent question was tying plant functional traits to climate. And in our system, we have two dominant grasses, black grama and blue grama. And black grama is a desert grass. And as you can see in this plot on the top of my screen, they're both foundational species, but the abundance of the black grama represented in the gray is encroaching on the area of the blue grama represented in blue. And now that we know that producers in our system do have responses to climate, and we were able to evaluate those using their functional traits, the next step was to use consumers and evaluate their traits to understand their response to climate. So I was looking at the small mammals of the Sevilleta. The 5 main genera that I focused on were in the 2 dominant families found at the Sevilleta in order Rodentia. They're Heteromyidae and Chrysalidae. In Heteromyidae, this consists of kangaroo rats and pocket mice. In Chrysalidae, mainly in the Sevilleta, consists of harvest mice, pocket mice, and paromyscus, commonly referred to as deer mice, but in the system, the most prominent paromyscus species are the cactus mice, pinyon mice, brush mice, and white-footed mice. I wanted to show you a little bit about our trapping methods and how we collected this data on the small mammal species. So small mammals are captured using Sherman live box traps, which are small metal boxes with a quick release door that closes when an animal enters behind the bait into the baited traps. And then they're placed in a web design out at our site. And a web is a wheel-shaped design with 12 spokes radiating out from the center spoke. And the inner traps are placed around 5 meters apart, with the outer traps being placed around 10 meters apart. And the LTR has 2 main sampling seasons, spring and fall, which coincide with peak green or greening up of vegetation that occurs when it warms up in the spring, and then again after the monsoon season. So therefore, the small mammal trapping occurred alongside the plant data collection. Our LTR staff scientists are experienced field research that are trained in a multitude of ecology protocols approved by IACUC in proper handling techniques for small mammals. And once the species are captured, we try to identify them to species or genus, tag them with an ear tag for an individual identification, check their sex and reproductive status, and then take morphometrics or measurements to help us with species identification. Another LTR data set that I was fortunate enough to help manage for the 2 years I was with UNM is a monthly small mammal trapping protocol that happens alongside the seasonal trapping. The advantage of this protocol is that they trap 9 months on average out of the year, depending on weather. So they trap every single month around the new moon schedule, which allows them to have the most capture rates because small mammals are more easily caught during the new moon schedule when they can evade predators with lack of moonlight. This site was selected particularly for isotopic analysis of small mammal diet to differentiate between carbon and nitrogen composition. In addition to all the same morphometrics and measurements we took on the small mammals for the seasonal trapping, we also took samples for the monthly small mammal trapping. And in addition, we would place the animals in this machine that's you can see in the bottom right of my screen. And this is referred to as a QMR, which is a quantitative magnetic resonance machine, and it's similar to an MRI. We would place the animals in these perforated tubes and put them in for a 3-minute scan so we could obtain body condition information such as % fat and lean mass and total water content. Finally, the last data set that I used for my research incorporated meteorological or permanent weather station data. These stations are throughout the Sevilleta and are located near pertinent study sites such as the animal monitoring. And they collect hourly meteorological data, which we use to determine climate over time, including that aridity index I've referenced. For the functional traits I mentioned, we had to go through literature and empirical data to obtain relevant traits for these small mammals. And the hope was to compare their individual climate sensitivities against these adaptive life history traits to predict the climate sensitivity. And we, like I said, selected these small mammal traits using empirical data from that seasonal small mammal trapping protocol, along with the monthly trapping protocol. And these traits can be broadly categorized as reproduction, diet, behavioral strategies, or body condition. So for the first part of my statistical analysis, it was to determine these climate sensitivity functions, or CSFs, for these individual species. As I mentioned, the aridity index was conceptualized to combat the fact that water availability in an arid desert ecosystem is driven by temperature, evapotranspiration, and precipitation. And we compared that SPEI, or aridity drought index, against small mammal abundance as our ecological response. Small mammal abundance was determined taking the individual animals caught, dividing by the number of trap nights in each sampling bout, and dividing by the kilometers squared area in which we sampled. Using those parameters enabled us to create these complex CSFs, and they involve interaction between climate mean and variance. And for the second part of the analysis, we were taking those complex climate sensitivity functions and comparing them against the functional life history traits. The traits that had significant p-values were selected as our top traits, and those indicated that they did correlate as predicting a species sensitivity. Of the top traits, those were then ranked to in value to determine which traits were the most important in predicting a species sensitivity to climate. So for this, I'm gonna use my laser pointing. For each species, we divided them by the two habitat types I referenced, the grassland and the shrubland. So because I was looking at 15 of the 23 small mammal species, we had 30 plots for each species. So rather than show you all 30 of those plots, I wanted to give you one example of one species, and then I will show you the total responses for all 30 of them. This species is one of our medium-sized kangaroo rats, and as you can see, their population abundance is much smaller in the grassland. They're found much more often in the shrubland when we do our small mammal trapping. So because the abundance is higher in the shrubland, it created a cubic response for their climate sensitivity function, which is a nonlinear response, which is what we were looking for. Because climate is nonlinear, we assume that a species climate response is also going to be nonlinear. I also wanted to show for this species, you can see on the x-axis that in the grassland, the species had a lagged response time of 12 months, whereas in the shrubland, they responded to our climate models within 6 months. And sorry, to cancel my laser. Technology, my apologies. Okay, well, I'm just going to proceed. So anyway, so this is a very good example of the different type of response curves that we were looking for. And now I'm going to show you the total number of responses. So like I said, there was 30 plots total. So to visually quickly show you what that would look like for each response curve, I did a total count of the 30 species and then created a bar plot so you could see that for each species, they had a different response, whether it be a linear response or a nonlinear response, which are the 4 bars on the right, which include cubic and quadratic responses. And so for these, we were looking to see when their abundance was going to be high. So on a linear response, this is when their abundance will be high. On a quadratic response, these two climate extremes show their highest abundance. In this climate extreme, or moderate climate, the abundance is high. And then here, they are both high and low. So these nonlinear responses indicate to us that these species are responding to climate variability. And this is an aridity index, so you can kind of visually see that for these 10 species, when their abundance was highest is when our drought index was low, and it was cold and wet for the climate. After we created those CSFs for every single species, then we went on to model the coefficient parameters from those plots against the traits. And of our 15 traits that we were looking at, 10 of them were significant in predicting a species response, particularly in the grassland. You can see for these 5 traits, the only trait that predicted a sensitivity was in the grassland for the linear CSF terms. And these were the other 5 traits where we had a few more responses. Again, we had strong responses in the grassland for the linear CSF terms and some responses for the quadratic and 1 in the shrubland. So I just wanted to give a little more background on these individual traits that were significant. So for breeding season end date, the breeding season end can range from spring to fall, as indicated with my icons here. And oh my gosh, I can't see my notes. So as you can see in the plot on the left in the grassland, for the end date, the earlier ending species include those deer mice, the Paramyscus, and the Dipodomys or harvest mice. And then on the other end of the spectrum, the later end dates for breeding season include the kangaroo rats and the pocket mice. So a little ecological background to explain this trend is that harvest mice, or the chryseidids and Paramyscus, they end their breeding seasons early because they have to prioritize breeding within a shorter breeding season window, as you can see on the right side of the screen. And so heteromyids, or the kangaroo rats and the pocket mice, often wait to reproduce until after the monsoon season has ended. And that's when the optimal growing conditions have been met, so they are able to reproduce. So they are a little more flexible with their timing and are less restricted to a specific breeding season window as the Paramyscus are. Additionally, the chryseidids must meet a certain body weight within this shorter window, breeding season window, before they can reproduce. So wetter conditions provide for more resources for them. So that results in higher abundance when the climate is cold and wet. Another trait within reproduction that was significant in predicting a species' climate sensitivity is the number of litters per year. So there are species that produce more litters of offspring per year are more abundant when SPI is high, the aridity index, which, as indicated at the bottom of the screen, is when the climate is cold and wet. And species that produce less litters of offspring per year are more abundant when SP is low or when it's hot and dry. So coinciding with the breeding season timeline, by having a flexible reproduction timeline for the kangaroo rats and the pocket mice, they may only have to reproduce 1 time per year. So they have a year to wait to do so. Whereas the chryseidids, such as the deer mice and the harvest mice and the grasshopper mice, they must reproduce as many times as they can within those restricted breeding seasons, but will reproduce more when wetter conditions are met because they are likely going to have more food resources available to them. For granivory is a diet consisting primarily of seeds and grasses versus an insectivorous diet or omnivorous diet consisting mostly of insects. So for these species, if they were more granivorous, they were more abundant when SPI was low, so when it was hot and dry, and less granivorous species were more abundant when SPI was high or cold and wet. So as I touched on, optimal growing season to produce resources is important to granivores, but can be offset with a stored food resource, while the chryseidids, the harvest mice and grasshopper mice that only rely on a variety of resources, including only a supplementation of seeds, they have to rely on what they can forage on. So if they do not have enough seeds to supplement their diet, they must rely on other major components of their diet. For insectivory, which is, as I mentioned, omnivorous diet consisting mostly of insects, the more insectivorous species were more abundant when the climate was cold and wet, and the less insectivorous species were more abundant when the climate was hot and dry. So as I indicated, omnivorous species that supplement their diet with seeds, but primarily feast on insects or other omnivorous prey items, will again be reliant on a wetter season to produce more insects and prey. So the seasons that they are productive provide strong resources in the form of prey for these omnivorous species, making them more abundant during climates such as this. For caching, caching is a way for some of these species to store their food either in burrows or tunnels, or shallow caches underground using their cheek pouches, usually indicative of the kangaroo rats, as you can see in the photo, or pocket mice, whereas the other species like the deer mice and grasshopper mice do not cache and store their food. Instead, they consume their food as soon as they forage for it. So for caching, the cachers are most abundant when SPI is low or a hot and dry climate, and the non-cachers are most abundant when SPI is high. Additionally, cachers benefit from climate variability, while non-cachers do not. So caching is the sum of all parts and the holy grail for kangaroo rats and pocket mice in these ecosystems, and it provides a way for them to outcompete the non-cachers during hot and dry climates. Additionally, this trait showed significance statistically because these species benefit from climate variability. This is likely because their caching allows them to be adapted to a hotter and drier condition, and they can respond positively to periods of variability, introducing more wet conditions in which they will also benefit, because then they can replenish their food stash from any resources that are produced during those wetter conditions. For fossoriality or burrowing, so all the small mammals in this community do burrow, but there's varying levels of burrowing. The larger kangaroo rats in particular are known for very deep and intricate burrows, whereas the harvest mice are known for just burrowing at the base of cacti in a shallow burrow. So for fossoriality, the deeper burrowers were most abundant when the climate was hot and dry, and the shallow burrowers, such as the harvest mice, were more abundant when the climate was cold and wet. Because of the deeper and more intricate dwellings, the species that are more fossorial may be able to escape from more heat and exposure than the other species, while the harvest grasshopper and deer mice may be more adapted to colder and wetter conditions because they have more exposure. Finally, for body condition, in the form of percent body fat, we saw that in the grassland in the spring, the higher variation in body percent fat species with this were most abundant when SPI was low or hot and dry, and they also benefited from variability. And variation in the fall, species with higher variation in their percent body fat were more abundant when the climate was hot and dry. And finally, for average percent body fat, species that had higher percent body fat were more abundant when the climate was cold and wet. So some quick background, because I am running out of time. For variation in spring body fat. The species that had the highest variation was the silky pocket mouse. If their body fat, it's beneficial to have higher body fat because it will allow them to increase their abundance. And this species is known to have highly individualized diets, which allows them to switch back and forth between a more specialized diet and a more generalized diet, depending on the boom years of their populations to minimize competition. So they are able to outcompete the non-cachers during a hot and dry climate. And so they will have more variable body weight. Very little variation in body fat usually indicates the species or individuals are not fluctuating within their weight. So species like a pinyon mouse that are highly dependent on their fat stores, particularly after a winter season and going into spring, will be more successful, so their abundances will increase in colder and wetter climates when their prey items are more abundant. For variation in the fall, the shallow caching, smaller of the kangaroo rats, was thriving the most under the variation in their body fat in the fall because they rely heavily on less nutritious but fallback food sources that typically sprout after the monsoon season and enable them to choose quantity over quality to quickly fill up their shallowly corded caches in multiple locations. Whereas the fatty species, known as the grasshopper mice, usually also the harvest and peromyscus species, will want less variation in their body fat going into the fall and subsequent winter season. By the fall, their dominant prey should have been present for months, giving them ample time to build up their fat storages to encounter hot and dry conditions in the fall. And then for average body fat species that have a lot of body fat, the fatty species are those that do not store food. Their abundance will increase when they have higher percent body fat because they were able to enter a winter season with higher fat stores. And so it'll increase their survivability over the winter and allow their abundance to increase in the spring after winter has ended. Finally, as I mentioned, we did rank the traits by importance. So I wanted to just give a quick visual so you could see that the most important trait was from body condition in the form of variation in body fat in the spring, granivory being still a significant trait, but the least important in terms of ranking. Some final take-home points from all of that data and technical jargon. If a small mammal's trait makes them more vulnerable to variability, climate can change the abundance of their species population, which then could ultimately cause a local extinction if it drops too low. And this would permanently alter the community structure if that species are lost. And overall, 10 traits predicted the species' climate sensitivities, with 2 traits making them more resistant to climate variability. Each species has a unique climate sensitivity, and their functional traits influence whether these individuals will be resistant or vulnerable to climate change. Species with the traits that made them more resistant to climate change were kangaroo rats, and in particular, the silky pocket mice seem to be the ecological winners when encountering drought and variability, while the chrysalids, the harvest mice, etc., are losing, which is in line with the observational trends we are seeing with their abundance. So some conservation strategies can be used to try and help protect these species if they are ultimately on the verge of local extinction. And so for conservationists to manage a healthy ecosystem overall, it can kind of help combat these issues that we are seeing with local extinctions. And that was it. So I will leave the last 10 minutes up for questions.
Awesome. Okay, so we do have a couple right now, and people and everyone in the audience, you have plenty of time to submit your questions if you would like. All right, so this question is, do you collect samples for DNA identification of species and individual animals?
Oh, that's a good question. So I was never a part of that. We are, there is a study I know that the monthly mammal trapping protocol is currently pursuing, which involves DNA barcoding of their fecal samples, which will help identify the plant species that they're consuming. Because as I mentioned, we do run their blood plasma for isotopic analysis of the carbon and nitrogen content in their diets. But currently that just gives us values of carbon and nitrogen and not specific species. So there are studies that are interested in trying to narrow down the plant species. And then as far as the small mammal species, no, I'm not aware of any current DNA projects to identify it. Right now, it's just observational studies and using morphometrics to get them to species.
All right. Okay. Thank you. So do rodent predators, coyotes, foxes, hawks, owls, snakes, respond to climate variation in the same fashion? How might this influence contribute to your results?
Yes, that's a really good question. So one of the trends that I was referencing in there was that the small mammals in the grassland ecosystem more than the shrubland ecosystem were having responses. And I know that for some bird studies, as well as the plant study that happened at the Sevilleta, and as well as the bees that are being studied at the Sevilleta currently, we're seeing that they're kind of responding to climate variability as well, and in particular in the grassland. And similarly to small mammals, like some species are more successful because they have functional traits that give them better adaptations to be able to endure the changes in climate. Mm-hmm. Hope that answers the question.
Yeah, I think it did. Let's see. Any other questions from the audience? Okay. Awesome. So if anybody does have any questions for Jessica, you can feel free to put that on your evaluation form that you're going to get after this talk, and I will relay those to her if we get them. Awesome. That was excellent, Jessica. Thank you so much.
No problem.
Oh, we did. Sorry.
Oh,
sometimes typing takes a minute. If some of these small species were to go extinct, what would be the larger effect on large predators?
That's a good question. So, I guess I'm guessing they mean within the rodent community. So, at the end there, I kind of blew through the final result, but like one of our smaller species, the silky pocket mice, it's probably the smallest in terms of weight and grams. They were the ones that are the ecological winners, actually. So keep in mind, though, that for those, that species in particular, one of their biggest predators are birds and rattlesnakes, or any kind of snake in that ecosystem. So it's not necessarily like the size of the mammal that's going to impact what happens to the predators, but it's a good question because, yes, if that's an important prey item for those predators, like if the silky pocket mice were the most vulnerable species and they were to go to extinct, then the snakes would lose their prey resource. So they would have to then maybe go to like larger rodents or supplement their diet in other ways. So any loss of any species is going to have a snowballing effect, an ecosystem effect on the community.
Definitely. Okay, awesome. So a couple of people saying thank you. Okay. What effect do you think the absence of prairie dogs in the Sevilleta has on the success of larger species like K-rats?
That's a really good question. Yeah, so the prairie dogs I know historically were present, and I know that U.S. Fish and Wildlife is working actively to try to reintroduce them. But I could definitely, I don't know too much about the relationship between them, but because they are both very fossorial. If anything, the prairie dogs are more fossorial. That might be why fossoriality ended up being an important trait that was allowing a species like a kangaroo rat to succeed, because their digging and burrowing is more successful and more active because they don't have to compete with tunnels and burrows of prairie dogs in the area. So by removing the prairie dogs, incidentally, often associated with ranching activities, might have actually helped the small mammals that are competing for resources with them. But again, the absence of prairie dogs probably had, a downward effect on prey items or predators or the ecosystem in general.
Awesome. Great. Okay, if climate change eventually moves the availability of seeds to different areas, maybe higher in elevation, how do you think this would impact species that have different total areas of distributions?
Sure, that's a really good question. So one of the traits we did look at was home range size. So a lot of these species, they're at the kind of brink of their range, both latitude and longitudinally, but also elevation, excuse me. And so, yeah, there are some of these species. I know the Paramiscus, which are commonly referred to as the deer mice, that are much more active and present in the mountain range next to the Sevilleta, the pinyon juniper. So yeah, if food resources were to shift and be more prominent in those ecosystems, then that might, prove for those species that are struggling down in this ecosystem where seeds are, more readily available for kangaroo rats and pocket mice, they might be more successful at a higher elevation.
Okay. Would the fire history, lack of fire for many decades prior to 1973, and the fire suppression on by Fish and Wildlife Service until the early 2000s have any interaction effect with rodent population dynamics?
Yeah, sure. I'm not as familiar with effects of fire, but I do know, yeah, Fish and Wildlife is actively suppressing fires, but also they actually did a controlled prescribed burn one year that I was working there. So yeah, any controlled burn, it's going to deplete resources, for probably that one season. But then as we've seen in, a lot of management of using prescribed and controlled burns is in the hopes of like restoring a system. It can be really beneficial. Particularly, I know that the Collins lab that I referenced in this presentation, they've been studying fire in the plains and grasslands in Kansas, and they know that fire is like extremely important in providing resources. And so the plants might actually do better after there is fire. So yeah, it's a good question. If they had been controlling for it, have the plants adapted to that fire so that they, aren't as plentiful at providing resources? I'm not actually too sure, but I know that fire is a really prominent ecological question and what the downward effects of fire suppression or having prescribed burns does to an ecosystem.
Mm-hmm. Okay. Are there studies of hantavirus in the rodent species at Sevilleta?
Good question. No, currently there are not. The most recent study of hantavirus that I'm familiar with was NEON, that I referenced, the National Ecological Observatory Network. But I think they have since in the past few years stopped studying hantavirus altogether because there was a big boom of hantavirus several decades ago, but currently they have not found, as far as I know, in recent years, hantavirus down at the Sevilleta. But it would be interesting if they would continue those studies again to see if they could track the change in that virology, because it is possible for it to return.
Mm-hmm. Awesome. All right, so I think that's the end of our questions. Very good questions. Thank you, members. Thank you, Jessica. Okay, so if that's all, I'm going to say goodnight to everybody. And thank you so much to Jessica again for sharing your knowledge with us. That was incredible. And thank you to everyone that tuned in this evening. If you are interested in seeing more peak programs, we do have another talk coming up on Monday, February 28th, which is an encore presentation of a mountain lion program from the Valles Caldera. So tune in for that if you would like. And yeah, again, thank you everyone. Jessica, you're getting lots of bravas. Good job.
Thank you everyone. Thanks for joining. Appreciate it.
Yeah, and
thanks for having me, Kristen.
Of course, thank you so much. So without further ado, we're just going to say goodnight, everybody. Look out for that survey. And I hope you have a warm evening inside, away from the cold. Have a good one.